Hey all, welcome to ShareTrek. This is Raj here. Today I have with us Dr. Eric J. Art, and it's my pleasure to introduce Dr. Eric Art as a distinguished professor in microbiology and immunology at Western University. Dr. Arts began his journey into infectious disease research during his studies at McGill University, where he explored the molecular mechanisms of antiretroviral drugs and HIV resistance under Dr. Mark Weinberg's guidance. Continuing his research at Case Western Reserve University, Dr. Arts dwelled into HIV-1 pathogenesis and evolution globally. Establishing a virology lab in Uganda, his team processed thousands of HIV samples, conducted drug resistance tests, and maintained a vital sample repository. Dr. Art's groundbreaking work extends to developing approved technologies for HIV and hepatitis C drug resistance testing. His commitment to vaccine development underscores his dedication to combating infectious diseases worldwide. Please join me in welcoming Dr. Eric J. Arts as we delve into his remarkable contributions to microbiology and immunology. Welcome, Dr. Arts.
Thank you, Raj. I really appreciate you inviting me.
Thanks for giving us your time. It's really appreciated by the entire audience of the channel. I just wanted to start off with the icebreaker. How is it going in Uganda? I believe you just reached there and you're doing something out there. So can you just give us an idea of what's happening, how it is like in Uganda for you?
Yeah, it's wonderful. Uganda is like a second home for me. I've been coming here for almost 30 years now, so I'm back and forth, and it's like a reunion every time I come back. But most of what we're doing is just continuing our work on HIV treatment and drug resistance. And right now, our real goal is to meet with people here to try to set up some clinical trials on the HIV cure approach that we are working with.
Thanks a lot. I hope you have a lot of success there and everything works in your way, Doctor. And let us start off with the first question. Can you please explain to our audience how the HLP therapy works in order to address HIV?
Sure. So it can be a little complicated in the approach, but really it's a very simple idea. And that is when an HIV infects an individual, it needs a T cell to replicate in. And that T cell is also involved in the immune response to HIV. So HIV has a tendency to replicate in T cells that it's also trying to fight. And when you start antiretroviral treatment to block the HIV, then what happens is occasionally some cells that are T cells, they become dormant in some ways. They provide memory to our immune system, and occasionally they pull an HIV that also becomes dormant or latent, as we call it. And that can live for years and years. Some estimate the half-life or the time it would take to eradicate that latent HIV is for 70 years, for example. So anytime you stop your treatment, the virus rebounds very rapidly and you start progressing again towards AIDS. So we realized that one of the things that we had some control over is knowing that HIV in its dormant state resides in those T cells that also recognize HIV as the bad thing. And so we said, well, what if we just make a virus that's HIV, almost identical to HIV, except that we've developed some tools to kill that virus, but they're genetic tools. And then we can see if that will tease out the last remaining virus that's in those dormant state. And in some ways, it's kind of tickling or getting that needle in a haystack, if you will. And specifically pulling it out with a strong magnet is like the way we like to describe it. So it's very targeted, the approach we're using. And in the end, we're just giving, potentially in the future, I hope, a patient a dead version of the virus they're already infected with. And in some instances, we hope that we can achieve a cure that way. So I hope that makes some sense.
Yes, it does. And that brings me to the second question. The promise of awakening dormant HIV so that they can be eliminated is absolutely exciting and scary at the same time. As while dormant infected cells are exposed, the viral load can spike and infect new cells. This suggests a viral control mechanism may be needed in parallel. While will it need to work with ART in order to constantly flush out dormant HIV and kill it so that we have a sterilizing cure in the end? I mean, deploying HLP in conjunction with some killing agent, like we heard about EBT 101, that they are testing it with a dormancy awakening agent. So is there something like that on the cards?
Yeah, so we're working with a number of different teams right now. A group at Harvard, well, really teams around the world, to look at how the HLP can be paired with different killing mechanisms. The verdict in my mind is still out, and that's based on the natural process of your immune system. So if we were like tickling these cells and not driving them into the normal physiological process, these T cells, then you have a problem because those T cells might be producing virus and those T cells might not be eliminated. In this case, the normal process is what we're using in antigen, in it's kind of a vaccine response. And when you activate those T cells specifically that have the latent HIV, those T cells have really two choices. The vast majority of those T cells will actually help the immune system, so help the immune system fight off HIV in this case. But eventually they have to die. Those T cells will go through what we call pyroptosis. And that T cell death means that it's carrying the virus to its death as well. And the other thing that those T cells can do is fall back into this memory state to be saved for future immune responses to HIV, for example. And so in a sense, we don't need as much of the response that might help in the killing of those cells. The other thing too is that in this approach, you're never taking the individual off of antiretroviral treatment. You would go through these courses of HLP treatment with antiretroviral drugs. So any virus that might be released would largely be killed. So but of course it can always establish a little bit of latent pool. So it's like a diminishing return. You have to treat we estimate maybe 4 times before you would eliminate the last remaining vestiges of the virus. Now, with anything, a cure may never be fully achieved. Okay, so I also like to really preface what we talk about when we talk about this. And but I think in many instances, a long-term remission would be something anybody would be happy with in the circumstance. Of course, a cure would be better. Now, I'm not ruling out the partnership with other agents. There are lots of great opportunities for specific killing. Well, I shouldn't say specific killing, but more generalized killing that could work. The specific killings that we could use to target these cells, it gets into now aspects of genetic engineering. So like CAR T cells and other approaches to really target the activated cells. And then you get into this costs situation that goes way beyond what the HLP would provide in cost for a cure. Yeah. So I'm really kind of hoping we don't have to go in that direction, because I'm here in Uganda. I'm not doing this in a sense because I want a cure that works around the world.
Yes.
And to make a difference, not just for the rich people that exist in our human population.
Yeah. I can see while you are talking, I can see the way your mind is working in terms of making sure that the cost benefit balance is there and wide applicability is there. So you have taken a very elegant approach, and it may take slightly longer, but it's the most viable. So that brings me to the third question I have. We are looking at SAV001H from the same stable in the same university, your colleagues. And it was an approach where you used a killed virus with a bit of modification in them so that it doesn't infect, the replacement of a few components. I believe the entire process was quite expensive and not scalable. I still think it may go for phase 2. I'm not sure. I don't have much information on that. How does HLP happen to be inexpensive and easily deployable? Can you please compare and contrast, if you may, so that we can appreciate the promise of HLP? Because as you articulated, your focus is not only on a durable remission, which is a very realistic goal, but also making sure that it is affordable to a vast majority of people in the world.
Yeah, no, that's a good question. And we were involved in the SAV-001, and that vaccine I think could have shown a lot of promise. And my colleague Young Kang did great work on it. But you're right. I mean, in that situation, you are looking at a very similar vaccine to the IPV, so the inactivated poliovirus, which again is a classic example of what the high-income countries utilize for polio vaccinations versus the oral polio vaccine, which is largely in the low to middle income countries. And it's because of the cost of production and then inactivation. So I know that Young Kang is working on other approaches using a VSV vector. We don't have to go into it. That is much more affordable in production. So and we support him and work with him on that approach. So with the HLP production, it's actually a very low cost production because the attenuation we utilize, or it's not attenuation, it's basically killing the virus, but it does so through genetic modifications. And when I say genetic modifications, it's not introducing new elements into the virus, which could present problems, but rather we're removing things. So we remove things that prevent the virus from replicating, but doesn't prevent us from mass production through recombinant cell production approaches. So, I mean, the cost is not neutral, just like anything. And small molecules will always be cheaper to produce than a vaccine product. But the difference here is, with antiretroviral drugs, you're taking them for the rest of your life. And even if you are in a low to middle income country with various programs, still costing, upwards of maybe $1,000 with treatment and care at least. So it might cost, it's hard for me to estimate, but it might cost, maybe $100, $200 an immunization, if you will, or a treatment. I'd rather use treatment because it's not necessarily a vaccine. But if you're doing that 4 times, still a cost associated with it, but in the long run, it's way, way cheaper than anything. And if you compare to the approaches that have been used for a cure now, which we have cured some people, but it requires complete myeloablation, transplants, and we're talking millions of dollars on occasion, not
That's right.
hundreds of dollars. Yeah.
And still it is the price point. Of course, we can't hold anyone to the price points because the way I look at so many therapies on the investment side of our channel, costs are always unpredictable because where you make it and other legal considerations that come into play. But it is still less expensive than some of the art medicine that is being subsidized by LDCs and developing countries. So it seems very inexpensive. So wishing the very best for that. And then moving on to the next question. So I understand that your team has completed preclinical trials. Is the next step going to be some trial on non-human primates, or will you be able to start phase 1 with human trials? And if so, when do you expect phase 1 to commence? So it's a compound question.
Yeah, no, that's a good question. So we have done some primate studies in the past, and what we found was that the model wasn't actually that effective. What I don't mean is not that the HLP, it was hard to evaluate whether it would work as effectively or not. There's a few reasons for that. And one is that you can only use sort of a chimera of viruses, that is HIV and the primate or the monkey equivalent, which is SIV. And by doing that, that hybrid isn't as pathogenic. But it's also the treatments at the time, it's gotten better now, weren't as effective as they are for HIV in humans. So this works very effectively, and that's a caveat, in preclinical analyses if a patient is controlled their viremia with treatment. So, and unfortunately, in patients that may have developed drug resistance and they don't have controlled viremia, this won't be effective. In fact, there's possibilities that would actually be counterproductive. So those are some of the reasons why primates were really problematic. And then through consultation with lots of people, my colleagues, et cetera, and based on what was appearing with other trials, it just made more sense to go to humans and after animal toxicity studies to make sure that the approach was safe. And again, in humans, it's a bit palatable because we're giving something rather inert, and also to individuals that are on treatment and that they're already infected with a virus that this is based on. So that's the approach we're going with. So far, we haven't had anybody come back and say, yeah, no, you can't go forward without doing primate studies.
Right.
The other thing is, which is more concerning for me, is that the primate studies, the fact that the model isn't perfect for this, in order to get us to the point where we can actually test the HLP, which would be a SHIV, a CHLP, because it would be the hybrid,
Right.
which we produced, it would take about 3 to 4 years to really get an assessment.
Right.
And I just think that in the interest of the population, I don't see a purpose of actually doing that.
Makes sense. Yeah. So when do you think you'll start off? Yeah, sorry. Please go ahead. No, please go ahead. Sorry.
Oh, I think you were asking, though, about human clinical trials. So right now, the biggest task we have is producing it at what we call GMP grade so that we can use it for human use. And then also funding is always an issue. I mean, we found certain sources, there's a lot more attention to provide support. And my focus is now to really find that support to do it in parallel in a low to middle income country. I don't see any reason why that shouldn't be done. And we can provide the same safety measures, the same clinical trial units in terms of professionalism and quality exist here, where I am, for example, in Uganda, than it exists in the US or Canada or Europe. So that's my focus now. We'll see how that goes, but I think there are interests from foundations and elsewhere that have reached out, and I hope that that will go forward.
So, doctor, in that case, right, when the time comes in, I would be very happy on behalf of the channel to put up a GoFundMe if you're open to those ideas, and put a video there explaining the initiative and putting a target. And we can see that if that is something that is allowed with your setup, the way your setup for the research. So we can always talk about that at a future date when you have thought through. And
Yeah, no, that we can talk about that. I mean,
yeah,
I think in this case, when you look at where it would help the most, it's going to be the hardest to get the funding. I hate to say that, but it's the way things are.
yeah,
And that public response will force the hand of the large foundations, I would say. So,
yes.
so yeah, I appreciate that.
Yeah, we'll do that. And as soon as the time comes in, you just give us the signal, we'll be ready for that. Now, that
Okay.
takes me to the next question. We have seen the clinical trial progress can be unpredictable. However, it is possible um, to get a rough estimate, I suppose, of your planned timeline for when phase 1 may start and potentially end, assuming that it's just the safety or testing and probably a little bit of efficacy.
Yeah. So right now we're targeting probably the fourth quarter of 2025 is kind of our hopeful target date when we would have enough product, finalize toxicity analyses, and then we would be ready, select sites, et cetera. There's been a large of interest from my colleagues in Europe and all over that, I've been willing to be a trial site. And I'm just, we're sorting through it. I'm not a clinical trialist, so that's the other thing is I want to make sure that we have the expertise on board to take that forward. And then I can, just assist where it's needed, but I'm not the individual to obviously do that. Then, yeah, so I'm sorry, there was a part of the question I was going to answer and I just forgot it for a moment there.
Oh, okay. So basically, the question was that is there a timeline that is planned when you might finish?
Yeah.
the-
Oh, I know what I was going to comment on. So the first part, the first phase 1 trial, which would largely be for safety reasons, it would clearly be mandated to be done in HIV positive individuals that are on treatment. So normally you wouldn't do that with a drug. You would do safety studies sometimes in HIV negative patients as well as positive patients for a new drug, first in class type of thing. And in this case, because even though we can prove, I feel without a shadow of a doubt that it's dead, and in fact, it's very similar in the way we kill the virus, if you will, than that's currently being used in gene therapy approaches. But saying all of that, I think no regulated board will allow for that to go forward in phase 1 trials in healthy individuals.
Right.
So, but that is somewhat of a benefit on a phase 1 trial basis because we would be immunizing very similar to what we would do in a phase 2 or 3 trial, or if it ever becomes a real therapeutic. So that will give us readouts immediately on its efficacy, in addition to the safety. So to me, it could move relatively quickly if it works, right? And that's always the big if.
Right. So I would assume that if started, I mean, it's very difficult, I understand, to give a start and end date for phase 1, but I would assume that it would take 24 weeks to 52 weeks to get all the results that you want in case once you start. Is that a reasonable assumption to make? Like,
Yeah, no, that's very reasonable. Yeah, 24, 48-week time points. I mean, hopefully we will have clearance to release the 24-week data before the 48-week data is up. And, as soon as you get, if you get good results on a 24-week data, then you can already start planning a phase 2 trial in
yes.
earnest. Like you would
Yeah.
plan it in advance anyways, but you at least could take it really forward seriously. So you could be in phase 2 maybe a year after you had started the phase 1. Yeah.
Okay. Now that brings me to the other question, which most of our audience asks always. One of the things our audience always asks is, how can we participate in the trial? So we have audience from across the world, and it's really heartening to see that your focus is more on the LDCs and developing countries. So is there a way they can volunteer? I know you'll have hundreds of people coming to volunteer, especially in Africa. So in which countries will the trial be conducted potentially? And if the audience wants to participate, most of them are HIV positive. Is there any way they can reach out to you, or are you soliciting those kind of requests? So
Yeah, I've received lots and lots of great emails, occasionally phone calls, and it is, and being in this field for I mean, almost since the pandemic started, it's it's really hard to hear stories, and I really, it's my life, right? And it's their lives. And the problem I face right now is I'm very optimistic that we will get into phase 1 trials, and that in phase 1 trials are just so small, right? You might see 50 participants
right,
in combined sites, or maybe 50 in each site. And then I think really from that point on, I really feel if it is successful, then you will see a very rapid response and expansion from funders to move forward
right,
with this. Would people, and I think the access would be, I mean, this is my opinion, the access would be almost to an extent that you would see similar to the COVID vaccines, right? That because I see it here, we do drug resistance testing all the time, and the dolutegravir-based regimens that were rolled out just before the pandemic, they're exceptionally good, but there are issues in association with that, and the biggest problem, especially in low to middle income countries, is drug supply. It's not always even adherence of the individual or access to the drugs. And so resistance always is a problem. And when individuals go down the resistance road, unfortunately, it's a slippery slope in many cases. And then you get to the point where something like the HLP would not be effective, right? So my opinion is, if the success of this is seen, then you will see UNAIDS and other agencies adopting this,
yes,
because it could spell eradication of the pandemic,
right.
not just but these are, and if it's HLP or if it's something else, I would be happy. I mean,
This is very
it's
early days,
yeah,
doctor. So my question is also a bit unfair. I'm just reflecting the aspirations of most of the viewers. I
yeah,
personally am not infected, but when I have been running this channel for the last 2 years, I have kind of put myself into their shoes, and I can feel the pressure coming from there. So that's why I asked the question. Thanks for
yeah.
being kind enough to ponder over it, given the fact that it's so difficult to answer that. But that brings me to the last question. I think, what is the, oh no, not last question, next question. What is your message to the millions that suffer from HIV? They always ask me, when will a cure come? So from your estimation, either HLP or any cure, what does it look like? When do you think we might have a functional cure or any kind of cure?
Yeah, so I think, the first phase of cure research, which was taking off the shelf, really chemotherapeutic type
Right,
drugs developed for other purposes, like the HDAC inhibitors or TLR7 ligands and various other approaches. They they it they did not do well in clinical studies. They had minimal effect, and it wasn't overly surprising because there were low dose treatments for months and weren't they weren't targeted. But over the last year or two, there's been some really good published findings of getting more and more specificity of where the latent HIV resides. And the reason why that's important is that you can develop these targeted approaches. I mean, we took a stab in the dark as to a targeted approach, and it looks pretty good, but there may be other ways of targeting these cells. And that's really the key, I think. And once we start targeted approaches, then we are getting closer and closer to a cure. Because that's really what stands between us and a cure is these last remaining cells that have HIV in it that can restart progression. And it can be effective that way. There will always be dormant HIV in an individual, even one that's cured. Even in the Berlin patient and other people that have been cured with these bone marrow transplants, they almost always have some residual virus that you can amplify. Not virus, but just remnants of the virus.
Right.
So, but that's not a problem. It's just a footprint of something that was there previously. So I think it'll work now.
So if I was to just condense what I understood, basically, we already have ART, which is killing all the viruses. We have the immune system, which when activated can target and kill the virus. And by crossing the last mile, which is activating dormant pools, which keep on reinfecting, we are progressively reducing the pool until there is so less remaining that it won't be a concern for the patient. And potentially, maybe in another 10 years, you would hope we cross that hurdle?
Yeah, I kind of, the way the science is going with pharmaceutical companies and and even our approach, once you get past preclinical development, it's not so difficult to assess the success of these approaches, right? The trials are rather short in length, and then you know if you have something that works or not. And as long as the safety profile is good, you can move several into phase 1 trials and then within a year know if they work or not. So it's kind of like which one hits the first effectively in phase 1 moves forward. And so that can be a process that if preclinical development is done on some of these therapeutics, which I know some pharmaceutical companies have, then it can move through that process at a reasonable pace. So somewhere between 5 and 10 years, I think is reasonable to say.
Okay. That sounds good. And this last one is slightly out of the topic because not only HIV, you also work on other areas. So this last question is for those in the audience with hepatitis B and HIV. So hepatitis B is a very interesting virus, the way I had seen it, part DNA, part RNA. I have read about ASC22 that is being used to target hepatitis B. And some people are testing it along with another drug called Shidamide to target HIV. However, your approach is HIV specific. I'll assume that HLP is only for HIV. I have seen that you've also been working on hepatitis. Are you currently working on a cure for hepatitis B, or do you have any plans to do so in future?
Yeah, so my research years ago, we did some work in hepatitis C, which is quite different than hepatitis B. And I mean, I know hepatitis B, but I don't really focus on research in that area. But I know the approach that you're referring to, and that is using like a PD-1 L1 ligand and that is an immuno-based therapy to kind of activate the T cells to target and kill something like an HBV infected cell. And the same approach can be used in HIV too. So that's one aspect that we've thought about and explored with HLP is to come in with an immunomodulatory drug like that turns back on the T cells to help kill whatever's there. The trouble with those is when you're on therapy and you're doing really well, and your viral load is undetectable, then those immunotherapies are not neutral in terms of side effects or even very serious side effects. So they make sense when your life is threatened with, for example, cancer,
Yes.
or you look like you have markers that you're going to approach cirrhotic liver or hepatocellular carcinoma or something like that with hepatitis B, for example. So those always have to be weighed in these circumstances. And unfortunately, the immunotherapies like that, they're an amazing discovery for cancer. And but they're a chemotherapy that still comes with cost
Yes.
in health. So I think a lot of approaches in use for HIV and cure had thought of about that approach in the past, but have kind of pulled away from it, because how do you convince a person that, yeah, maybe there's a small chance you become cured, but there's also a small chance that you may even die from this treatment. So, it's the same thing with Hep B in that respect.
So, doctor, it brings me to the next question. There are macrophages and dendritic cells who may be harboring HIV and are dormant. So what are your thoughts on those cells from the HLP perspective?
Yeah, and this is a common question, very astute question from many astute, very enlightened researchers. And so there has been reports of dormant HIV in myeloid lineages like macrophages, dendritic cells, and it's definitely there. The proportions of the latent pool in those populations are very low. So that's one thing. And we're going to get into the weeds a little bit. And one of the things is, so we have been thinking about how we might deal with that. And it's difficult because those different macrophage populations exist all over the body, not just in lymph nodes, for example, where the HLP would target for the most part. But one of the things we are doing in that presentation to the T cells of HLP to the T cells, it involves a dendritic cell or a macrophage or a B cell or some approach like that. So those cells, like you mentioned, have the receptors, so HIV can enter those cells. So what we've done is put in what is an RNA adjuvant into the HLP. And this is like, I would say this is HLP version 2.0. And the reason why we did that was we've taken the virus genome nucleic acid out of the HLP. But what we found is when we did that, there was a little bit of a loss of its potency. I mean, very minimal, but still. So we said, well, what if we put in an RNA into that HLP that is an immunostimulatory RNA, but only within the targeted cells?
Yes.
And because those cells are what takes it up for antigen presentation, when they do that, they get shocked by an RNA adjuvant. And so that would potentially drive out the latent HIV in those cells. So that's one approach, but I'm not too sure how much it's going to hit, but all of the myeloid cells, but at least it's doing something. Yeah.
Like a Trojan horse.
Yeah, very much like that. Yeah.
So, Doctor, that brings us to an end of all the questions. Thank you so much for taking time out of your busy schedule. And so from me and all our audience, it's really significant that you chose to take the time and talk to our audience, because while a cure is ultimate requirement, patients have apprehensions and an eagerness to know any detail they can get in order to manage their expectations and psychological disposition. Your interview today has brought hope to many people across the globe and a potential audience that may view it at a later date. So on behalf of the channel, all the viewers, and from myself, I would like to thank you for that. And we'll watch your progress eagerly and stand ready to support your initiative in any way possible if a need should arise. And we are capable of doing that. And hope you will meet with us in the future when you achieve milestones, or if you want to make any appeals. So thanks once again.
Thank you very much, Raj, and to Share Trek. And it was a pleasure to be here from Uganda, nonetheless. Yeah.
Yeah, that's a very nice place to start off our initial meeting with, and hope great things come out in future.
Thank you. Okay, bye then.